Thank you, Mark, for warming up the room. So the biggest question, of course, is for Gustav that everyone has in its mind. But the other question, of course, is also, how does all of this work in the wild? So what does it work in a routine lab? What can we already deliver with that? And I'm very excited to be presenting for the first time the work that we did in the past, less than 6 months. First, a brief introduction on Hartwig Medical Foundation. That is a non-profit organization, and we're actually doing routine whole genome sequencing-based cancer diagnostics for various hospitals in the Netherlands. Besides that, we actually develop open source analysis software to basically analyze this genomics data and to drive data-driven decision support tools in the hospital for oncology. So these tools are used for two reasons. One is to generate a patient report with therapy guidance that goes back to the hospital, and the other part, with the consent of the patient, is to collect this information, enrich it with clinical metadata, and that is to basically build a learn and care cycle where with the data of the patients of today, we improve care for tomorrow's patients. So that's the objective of our foundation. We do this whole process under ISO accreditation, and the average turnaround time is about 6 days. What is hampering us to get towards routine implementation? Of course, it's new technology and it's a lot of data, but typically we're hampered by two main things. And one is basically, this is too costly, and it takes still too long. So this is why we're always excited to explore new technologies to see if we can address these challenges. So we are very happy that we could team up with Roche on their new sequencing technology. This is a bit of the timeline of the beta test that we did. You basically see the prototype equipment there, not as nice and shiny, but nice orange, which is the Dutch national color for the expander generation. So basically in October, November, so this is installed in September 2024. So we started off with some test runs and optimization of our software, because I do want to stress that we try to be as independent as possible from Roche, especially for the data interpretation, because we feel every new technology needs to be assessed under high scrutiny to see whether it's on par with what we're able to do already in our controlled clinical accredited environment. Actually, we did the first production run in December, and actually what I will be talking about is work and data that we actually generated at the end of January, which is done on 30 patients and some COLO duplicates. Ewout and Joris, I want to acknowledge them specifically because they really got the equipment very quickly working and under control in the lab. And also Thomas and Peter, very instrumental as well to basically adapt our software to be able to work with the data coming from the new sequencing equipment. I don't have to say anything about this whole process. Mark already explained that. I will talk about 30 tumor normal pairs and a COLO cell line control in duplicate. Be aware we do tumor normal pairs and we sequence actually per sample, per patient. We sequence 4 genome equivalents. So this is a whole experiment encompassed in total about a bit more than 120 30x genome equivalents. And that was spread over 8 4-hour runs. That was done on a single chip, and the sequencing was done over a couple of days to generate this data. This was still done in a manual way, but Ewout and Joris also succeeded in automating this whole protocol and going from sample to data analysis, or to data production on 4 samples could be done in 24 hours in an automated setup on Biomax. So meaning this is a versatile protocol that can be easily automated. Okay, what do we then get? Let's walk you through the different steps from raw data to the complete analyses. This is basically what we typically get. So what you see in these graphs is the tumor and normal data, and this is the aggregate information of the 32 samples. So on average, we've got 150 gigabases of data for normal and 410 gigabases for tumor. The spread of coverage is very good, meaning like there is not much variation between samples. This coverage, this is duplex data, but it's derived from 2 different setups. Mark just mentioned that full duplexes and partial duplexes. In our setup, on average, the duplex rate, the full duplex rate is about 70%, and the N50 for these full duplexes is about 190 base pairs. The partial duplexes have a slightly longer insert length, but you see that the termination of the duplex part is sort of random over the stem. and that basically results in a duplex, average duplex length in N50 of 110 base pairs. If you then convert that to mapped coverage of these reads, so that was the raw data, this is now the mapped data, we basically see that we get about 105x coverage for the tumor and 47x coverage for the normal sample. And for the rest of the presentation, I will keep comparing this with our production data. So these samples, so these are patients that were also analyzed in our production setup on that's currently running on NovaSeq 6000. And these patients had identical tumor coverage and slightly lower normal coverage. Okay, let's first look at coverage. GC biases, I think this, don't look at the height of the curves, that just coverage differences, but I think you should focus on the left and the right of these curves, where you basically see that there is differences in GC biases, where you basically see SBX coverage dropping off earlier than Illumina at low GC, but at high GC, the performance of SBX is much better than Illumina. So this results, of course, in dropouts, which you can actually see some IGV views. You see dropouts in SBX in AT-rich regions, but the same thing happening for Illumina in GC-rich regions. And if you look at the overall numbers, the coverage genome-wide is not much different. There's, of course, a second effect, and that's the reads are longer. Compared to 2 times 150, you have 300 base pairs to map something in a genome, and also you have a mate which is slightly further away from the other sequence, from the other mate. So you get lower mappability due to the shorter reads. You see that in the white parts, the white reads have a map quality of 0, so they can also match somewhere else. You actually see that this region in this screenshot here already has more challenges in Illumina data as well, but you see that due to the shorter reads, this region starts dropping out completely with SBX data. However, if you look on where this is happening, and this is mainly outside the genome in a bottle high confidence regions, so if you correct for that, and you just look at the high confidence regions, you basically see almost identical performance for both platforms in terms of coverage. Okay, that's coverage, that's the basics, that's where you start. The next step is basically variant calling. We did this with our package of tools. So this is a package that we use for in routine diagnostics, and it's not just variant calling, there's also HLA typing, viral detection, all these type of things that can be relevant for the clinic and for a cancer patient. You see the features here. You can use all different types of data as input from FFPE, from panel, etc. But one of the things that how we designed this software is also to make it platform agnostic. So it should run with every platform. And the key tool in this is Redux, and that's a pipeline component that deals with platform and sample specific characteristics. And that feeds into the small and structural variant calling tools. And we basically took us a few months basically to optimize this tool to also work with the SBX data, which has its own characteristics, and the rest of the tools of this pipeline were not modified, so meaning they should all work once you have the Redux tool properly working for this SBX data. So what this Redux specifically does, it actually determines empirically the error rates to enable this high sensitive somatic SNV and indel detection. So that's not a machine learning that you do once, it does that for every sample that comes in. So an FFPE sample has a different error profile, and that's taken into account for the variant calling. And this is basically what it looks like for SNVs. It's a trinucleotide context error rate that we determine based on a few % of the genome that we survey. For errors compared to normal reads. And this actually allows us also to calculate the empirical error rates from the different platforms. On the SBX platform, there is no different quality bins like there is for Illumina, but you have simplex and duplex reads. And if you apply this tool, so this is completely independent of any of the tools that Roche uses, we actually find the Phred score for simplex for SNVs of 30, and for the duplex reads, it goes up to 50. And if you basically compare the two platforms and consider all the data, we actually do see that the empirical platform SNV error rates are lower than on the Illumina platform. Does that matter, this 1.7-fold, or the way, whatever way you calculate it? I'm not going to argue about that or make pleas about that. It's eventually, I think everything above Q30 is basically suited for the way of we do variant calling in this for somatic variant identification. Okay, then basically what do we find? This is basically a COLO sample, and basically here you see the challenge that you have with somatic variant calling. It's not a bin of 0, 50%, 100% variant allele frequency. No, it goes all over the range from 0 to 100%. And basically at the bottom of the graph on the left corner, that's basically where noise starts meeting real signals. Actually, it's mixing. It's not even meeting each other. It's basically a mixed part of signals. So it's difficult to interpret these graphs. But basically what you see in this case, this is the raw data, you basically see a high degree of shared variance. So where do the private variants come from? Well, about 20% is along these axes, no coverage in one or the other platform. That explains already a big chunk of the privates. And the other part is basically, if you look at the private variants and look at variant allele frequency in the other platform, you basically see that they're all very low variant allele frequency. So we're actually looking at stochastic effects around just meeting the threshold for variant calling. So a simple thing to get this out of the equation is just to make a cutoff of 10% variant allele frequency and then do the comparison. And then you basically see that you get sort of two identical numbers with the symmetrical private profile, again, around the new cutoff that we defined. We believe these private variants are real variants when you look at their mutation spectrum. This is about patients. You see the same pattern. You see very high concordance between a number of variants that are called in these patients with very low rates of private variants across these 30 patients aggregated. Indels, same story. We basically see, well, this is the COLO again. Please note that there is only very few indels in COLO. This is a melanoma sample, but there is also very clear that you don't get high rates of noise because of different specific error profiles. Patients, same story for the indels. A lot of shared variants. And very few privates. The only privates that we do see are happening mainly at T homopolymers, which you basically see here. T homopolymers are much more abundant than C homopolymers in the genome. And you basically see above length of 6 or 7, you start seeing more Illumina private variants. We don't know if they're false negatives in SBX or false positives in Illumina. I just want to make a note that we don't use a PCR-free library prep in this setup, so these could also very well be PCR artifacts. Structure variation, copy number alterations. This is still work in progress. This is a first screenshot on COLO 829. You basically see on the inner circles that the copy number profiles are highly similar from both platforms. Structure variance is also looking quite promising. But we need a bit more tool optimization to get this completely done for all the patients as well. What is the impact of private variance? Actually, if you nail this down, we seek private variants across these 30 patients. But if you start looking at how many are in exomes and how many are actually in actionable genes, clinically relevant genes, there are only 5 variants left. And they're spread over the 2 technologies. And the reason for missing them in one or the other technology is actually quite variable, as you can see on this slide. And we basically come to the main conclusion that no clinical impact, that there was actually no clinical impact due to platform differences. I'll skip over the summary because this will be repeating what I just said and just come to the end conclusion that we feel that SBX provides a very promising alternative platform for rapid whole genome sequencing-based cancer diagnostics. Thank you for your attention.